Hello, everyone. Good morning, good evening, good afternoon, depending on your time zone. Welcome to this month's edition of webinars organized by the next generation vaccinologist of the International Society for Vaccines. My name is Omolara Baiyegunhi, and I'm your moderator for today. The next generation vaccinologist group of the ISV organizes these webinars monthly to showcase the research of early career investigators as well as well-established researchers, and also talk about career trajectory and many other topics that are of interest to early career researchers in the organization. So we have exciting topics by two very interesting presenters for today. But before we get into these presentations, I'd like to give a few announcements from the next generation vaccinologist group. So we would like to take this opportunity to encourage every one of you attending this webinar today to join the International Society for Vaccines. So you can join by visiting the website here that is displayed right here on the screen. And there are many benefits for you joining, especially as an early career researcher. You will get the special discounted registration membership fee of $35 US. And you also have discounts to register for the annual Congress. And then you can have access to career mentorship, as well as many other privileges that are reserved for members of the International Society for Vaccines. Importantly, you would have great opportunity to network and collaborate with experts in the vaccinology sphere. And you also will get discount to publish your work in the Human Vaccines and Immunotherapeutics Journal. We also have an announcement for our upcoming webinar, which will take place in January. And we are excited to announce that we will be having a talk by the winner of the Bright Sparks session during the ISV Congress that happened this year. And that is Alexander Sampson from Oxford University. We also will have the CEO and co-founder of Next Wave Bio, Manon Cox, talking to us about entrepreneurship. So please look forward to our webinar in January. To join our Next Gen Vaccinology group, you can scan this QR code on the screen. You can join our WhatsApp group because we have up-to-date information in that WhatsApp group. You can also follow our LinkedIn page by scanning this QR code. Importantly, please send us an email on nextgenvax with the double C @gmail.com. We look forward to receiving your emails. Just a few housekeeping rules before I introduce our speakers for today. Please stay muted until the Q&A session, where you can indicate by raising your hand and you will be invited to ask a question. Alternatively, you can type your questions in the Q&A box at any time during the webinar, and we will read out the question or will give you an opportunity to ask your question. So now to the business of the day. I'd like to introduce our first speaker, Dr. Ebony Gary. Ebony is a senior postdoc in the laboratory of David Weiner at the Vaccine and Immunotherapy Center of the Wistar Institute in Philadelphia. She focuses on the use of nucleic acid encoded adjuvants to enhance and direct vaccine-induced immunity. She's focused on understanding the molecular mechanisms of vaccine failure in special patient populations. And she received her PhD from Drexel University College of Medicine. We look forward to this exciting talk by Ebony, where she'll be talking about peripheral co-delivery of Plasmid Encoded Mucosal Chemokine CCL27 enhances mucosal immunity and supports protection from heterologous SARS-CoV-2 and H5N1 influenza virus challenges. So I'll hand over right now to Ebony to share her screen and give the talk.
Thank you so much for that introduction, Omolara. Can you see my slides?
Yes, you can put them in presenter view.
Great.
Perfect.
Thank you so much for inviting me to share some of the work I've been doing in the Weiner lab with everyone today. Today, I'm going to talk about how the co-delivery of plasmid encoded chemokine ligand 27 can enhance mucosal immunity and support protection from heterologous respiratory virus challenge in vivo. So the Weiner lab really focuses on the design of DNA vaccines, DNA antigens, and also RNA antigens now, so really nucleic acid vaccines. But a major focus of our lab is the design of genetic adjuvants, which we call molecular adjuvants, that we can co-deliver with our antigens to enhance and direct vaccine responses. And we've demonstrated that we can co-deliver chemokines and cytokines like IL-12 in both preclinical and clinical models to enhance T cell function, CD8 T cell function, to enhance antibody function. We've also demonstrated the ability to co-deliver DNA-encoded protein molecules that have enzymatic activity, which could enhance both germinal center function, T cell function, and promote long-lived antibody responses in vivo. But today I'm going to focus on the ability to co-deliver chemokines to direct vaccine-induced responses to specific anatomical sites like the mucosal surface. And in this case, I'm really going to be focused on the lung mucosal surface because I'm going to be talking about SARS-CoV-2 and influenza infection. And so I wanted to start by saying that peripheral vaccines typically don't induce responses that are robust at mucosal surfaces, and that's because peripherally activated lymphocytes can't access those mucosa-associated lymphoid tissues or MALT tissues. And the reason that they don't have this access is because MALT access is mediated by the expression of specific chemokine receptors and adhesion molecules that allow cells to traffic into those places and potentially reside there. And one of those chemokine receptors, chemokine receptor 10, which is the one I'm going to focus on today, is expressed exclusively on IgA positive B cells at mucosal surfaces and barrier resident T cells. And so that's T cells at mucosal surfaces, but also T cells in the skin. And because of that, one of its ligands is named cutaneous T cell attracting chemokine, CTAC. It's also called chemokine ligand 27 in the standard chemokine naming scheme. I'm going to call it CTAC for the purpose of this talk. And preliminary studies from our lab had demonstrated that when we co-deliver CCR10 ligands with DNA antigens, we could have a positive impact on SHIV challenge outcome, so on a mucosal challenge outcome. And so we wanted to test this first in the context of SARS-CoV-2 early on in the pandemic. And to do that, we're immunizing mice with our DNA plasmid that encodes the full-length spike of an early sort of parental spike, a Wuhan-like spike, or we're giving two plasmids simultaneously, one encoding spike, which I'm calling PS, and the other encoding mouse CTAG. So I'm calling that group plus PCTAG. And in this case, we're giving two doses separated by 3 weeks, and we're looking after the second immunization for responses. And I'm just showing you responses in the lungs by interferon gamma ELISpot here on the left and by intracellular cytokine staining on the right. When we take the lungs from these immunized mice and we do our interferon gamma ELISpot assay, we see that the CTAC group has significantly increased numbers of interferon gamma secreting cells in those lungs. And I think it's really beautiful when you actually look at the ELISpot, you can see that those increased frequencies of interferon gamma secreting cells are really across the full length of these matched spike peptides. And when we take those cells and do intracellular cytokine staining to ask what is the phenotype of those CD8 T cells in this case that are secreting interferon gamma, we see that we see a significant increase in interferon gamma secretion from CD8 T cells that are alpha 4 beta 7 positive. And alpha 4 beta 7 is an adhesion molecule that marks mucosal resident T cells. So we know that these are CD8 T cells that are going to live in the lungs of these mice and secrete interferon gamma when they come in contact with spike peptides. And so that suggests to us that we're really promoting a strong CD8 T cell response, specifically in the lungs of these animals, even though we're delivering our vaccine at a distal site in the periphery. When we ask about antibody responses at mucosal surfaces by collecting bronchoalveolar lavage fluid or cecal extracts from our mice, so that's a further distal mucosal site, and looking for IgA, so this is spike-specific IgA, we really only see spike-specific IgA in bowel of animals that receive CTAC and in the cecal extracts of animals that were co-immunized with CTAC. Similarly, when we take those samples and ask if they can neutralize spike pseudotype viruses, we really only start to see neutralization in the bowel in the presence of CTAC, and it's significantly enhanced in cecal contents from mice that received pCTAC. And what's really interesting about these really strong responses at the distal mucosa is that to collect those cecal extracts, we have to take out the full length of the intestines. And what we notice right away is that in our CTAC co-immunized animals, we start to see an increase in these Peyer's patches along the length of the small intestine. And Peyer's patches are isolated lymphoid follicles that can arise along the length of the intestines. And so this suggests to us that we're promoting a sort of de novo mucosal antibody response at this distal intestinal site, which is quite striking. And so we wanted to ask if these increased mucosal responses really had any impact on SARS-CoV-2 challenge. And we started these experiments in the beginning of the pandemic. And by the time we had this really robust data and we're thinking about challenge, the challenge in question was going to be Delta virus. And so I'll remind you that this is not matched to my antigen. So I'm giving wild type spike alone or wild type spike plus CTAC to these ACE2 transgenic mice. I'm giving 2 doses separated by 4 weeks in this case. And they rested for about 100 days before they were intranasally challenged with this Delta variant, which is wildly different from the parental spike. And so what we saw is we expected for our animals that received wild type spike alone, which is in gray here, we see about 60% protection from mortality in those animals. But our CTAC co-immunized animals were 100% protected from mortality. And when we looked at weight loss, as we expected for our spike-only immunized animals, we saw significant weight loss, even though 60% of them will survive. They were quite sick. But our CTAC co-immunized animals never lose any weight throughout the course of the challenge. And perhaps the reason for this is really evident when we looked at SARS-CoV-2 induced pneumonia in the lungs of these mice. So in our EpiVax animals, we see evidence of pneumonia, lots of perivascular infiltrates. And when we stain for virus nucleocapsid antigen, we see lots of brown staining in the lungs, suggesting ongoing infection. In our spike-only immunized animals, which again are sick, but many of them will recover, we again see SARS-CoV-2 induced pneumonia. We do see a significant decrease in nucleocapsid antigen staining, suggesting that there is some overlap in this protection against Delta challenge. But in our CTAC co-immunized animals, we see very minimal evidence of SARS-CoV-2 induced pneumonia, and we can't detect any nucleocapsid antigen, suggesting that we're really mediating this protection at the mucosal surface with this peripheral unmatched vaccine, which is quite striking and hadn't been reported before. Unfortunately, because SARS-CoV-2 isn't going to be going away anytime soon, we have the opportunity to continue to test how far we can push that breadth with this sort of mucosal immunization scheme. And so in this case, I'm giving wild type spike again or wild type spike plus CTAC, and I'm challenging animals with the Omicron BA.2 variant, which is actually not quite that lethal in mice, but we can measure viral loads and ask how much virus those animals are carrying after challenge. And we can see that by day 4 post-challenge, our CTAC co-immunized animals have undetectable viral loads in their lungs, suggesting that we're, again, mediating at least viral control for this, again, unmatched virus. And so we went back and did that again, most recently with XBB 1.5, which again is not lethal in the K18 model. And we see a similar trend that our CTAC co-immunized animals just have significantly lowered viral loads compared to wild type spike immunized animals. Again, suggesting that we're really promoting a mucosal protection that is broad enough to protect against these unmatched variants of concern. And so that's really exciting, but there are other respiratory viruses where changes in the virus are important for the context of continually developing efficacious vaccines. And one of those respiratory viruses is influenza A viruses. And so they are an ongoing public health threat, but they also represent the potential for new zoonoses to humans. So annually, there are about 1 billion flu infections, 3 to 5 million severe cases, and up to 600,000 deaths. And this season, there have been more than 25,000 flu deaths. The other influenza that I'm going to talk about today is highly pathogenic avian influenza, or H5 HPAIs, which cause sporadic outbreaks among domesticated and wild bird populations. And the incidences of outbreaks and zoonoses have been increasing since 2005. There is a current large outbreak ongoing in North America that's affecting cows, birds, and to date, there are greater than 50 human cases. And when we look back at H5 zoonoses and look at the numbers of infected patients versus the numbers of fatalities, we calculate a case fatality of about 52%, which is quite striking. And so we hypothesize that if we could mediate mucosal protection with our peripheral vaccine by co-delivering this chemokine, maybe we could enhance heterologous protection in the context of influenza A viruses, both seasonal and potentially highly pathogenic avian influenzas. And so our first pass was with this sort of lower bar of seasonal H1N1 virus. And so in this case, I'm giving a California 09, a seasonal H1HA plasmid encoded antigen, or I'm co-delivering it with CTAC. And I'm just giving a single dose and looking at T cells by ICS at day 14 post co-immunization. And you can see that our CTAC co-immunized animals have an increase in the frequency of CD107 positive, interferon gamma positive CD8 T cells in their lung, again, confirming that we can see this CTAC mediated CD8 T cell effect in the lungs of mice. We also have the ability to challenge mice with H1N1 in our lab. And I should back up and say that HA antigens are quite immunogenic and quite protective. They're a great vaccine. And so our vaccine, in this case, we gave a single dose again with or without CTAC, and we challenged these animals at day 14. Our HA-only immunized animals lose a little bit of weight, and we see about 90% protection, but our CTAC co-immunized animals never appear to lose weight, and 100% of them survive challenge, suggesting that even in this model, we could have an impact on both morbidity and mortality. And so we decided to try and transition that to the higher bar of the highly pathogenic avian influenza viruses. And so in this case, I'm giving the HA of an older clade of H5N1. It's called clade 2321C from a zoonosis that occurred in Cambodia in 2023. Or I'm co-delivering that HA with CTAC, and I'm giving 1 or 2 doses. This is the 1 dose data, and we're challenging those mice heterologously with an unmatched clade. This clade 2344B is the clade that represents the circulating strain in the current outbreak. And what you can appreciate from this challenge, where we're challenging these mice with 1 TCID50 of virus, that our naive animals are unprotected and completely succumb to infection by 5 days post-challenge. So it's quite a high bar for lethality. Our immunized animals, when we immunize with the HA alone or HA plus CTAC, appear to lose weight, even in the CTAC group, even though they do seem to be losing less weight. And when we look at viral loads in those lungs, we really only see a difference between animals that were co-immunized with CTAC and those that were given our empty plasmid, our PVAX, suggesting that the addition of the CTAC is helping to lower viral loads. And so we went back and we looked after 2 immunizations again. And really, again, that the striking difference is in the viral burden that these animals are carrying in their lungs. So when we co-deliver CTAC at 2 doses, we see a significant decrease in the amount of virus in the lungs of these mice. And again, this is a heterologous challenge, so unmatched to the HA that we're giving. And so we're quite happy with this data and excited to try and go back and do both homologous and heterologous challenges with other HPAIs. And so in conclusion, I've shown you that the addition of mucosal adjuvants to suboptimal SARS CoV-2 antigens can confer 100% protection against emerging variants of concern. For the Delta variant, this was the first report of 100% protection with peripheral delivery of a parental spike antigen. And we have since extended that to Omicron subvariants, and we have studies ongoing to continue to try and extend that to further Omicron subvariants. And also in the context of influenza, CTAC co-delivery enhanced protection from challenge, 100% protection from H1N1 mortality, and significantly lower viral loads in the context of highly pathogenic avian influenza challenge models. And with that, I will have to thank lots of people. It's a team effort in the Weiner Lab for both of these projects. Specifically Nick Tercy, Elizabeth Parzich, and our technicians, Casey and Corey Livingston. I would like to thank everyone in the Wistar course, especially the animal facility and the genomics course. the Patel lab that does our H1N1 challenge. At the Public Health Agency of Canada in Darwin Kobas's lab, they do our H5N1 challenge and our SARS CoV-2 challenges in containment, which is really critical to this work. And I'd like to thank all the funding sources involved in this work, including the NIH and Civics for the flu work. And with that, I will take questions.
Great talk, Ebony. Thank you so much. That was interesting. We have one question in the Q&A, but maybe I can start with a question of my own.
Sure.
And I was wondering if in your model, you were able to look at durability of the IgA response and yes, and what did you find?
For SARS-CoV-2, though the Delta challenge is out to about 120 days post my last immunization, and we bleed those animals before we ship them for challenge. And what I can get if I have to ship the animals alive are IgA and IgG in the serum, and I don't see any differences in peripheral IgG or IgA. What I can't get when I ship those animals for challenge is BAL, but we've done some match studies where we've rested animals for 100 days and euthanized them to do T cell responses at that time point. And while everything is lowered, there's still a CTAC-induced enhancement of both T cell responses and antibody responses in the mucosa, which is quite exciting.
Okay. Thank you for that. So we have two questions from Anke.
Sure.
And Anke says, thanks for your presentation. And they are wondering how you envision CTAC upon peripheral injection of the plasmid to work in the respiratory system.
That's a really great question. And people ask me that a lot. What we think is happening, which has been really hard for us to pin down molecularly, just technically, what we think is happening is that when we give our antigens, the cells at the site of injection are making antigen, but they're also making our chemokine. And so when cells traffic there and encounter antigen, they're encountering it in an environment full of CTAC. And so we think that we're causing sort of an autocrine increase in expression of the receptor on the surface of those cells. So now those cells express more CCR10, and then at steady state, they should use that CCR10 on their surface to home back to mucosal surfaces. So that's how we think that we get them there. We've done some studies in CCR10 knockout mice where we lose the effect. So we know it's receptor dependent, but it has been really hard to track CCR10 positive cells. So if anyone listening has a suggestion for that, we're very open to trying to physically see those cells going where we think they're going. That's a really great question.
Okay, great. And then you have another question from Anki as well. And they say, in the heterologous H5N1 challenge experiment, it seemed as if the mice immunized with the HA-containing plasmid only showed less weight than those that also got the CTAC. What are your thoughts about that?
Yeah, that's a really good question. I have a challenge ongoing right now, and we've been in detailed talks with our collaborators. So those viruses are super lethal in mice. And so that in that challenge model, we send 10 mice. And at day 5, they will euthanize half of them to do TCID50s for us and keep the other 5 to do survival and weight loss. And we just think it's not powered enough for us to really see those trends. And so we've gone back with 15 mice per group so that we can keep 10 mice for weight loss. But they think that weight loss will always be a thing that we see, that we'll always see, even if mice don't hit sort of terminal euthanasia criteria, that they'll always lose a striking amount of weight. They say the animals are quite sick. So we're interested in trying other H5 challenge models. There are modified viruses, more mouse-adapted H5 viruses that might be a better representative of what that virus is like. But yeah, it is sort of a unpredictable challenge model.
Okay, let me quickly ask a naive question for those of us not used to studies. So you measuring the weight loss as a proxy for disease severity?
Yes.
Understanding. Okay.
Yeah. So if the mice are sick, they will eat less and lose weight, but they don't really have sort of human patient symptoms, right? They won't have fevers, but we can see weight loss. Sometimes people will measure things like lethargy, but there isn't really an overt sort of clinical correlate for the way humans are sick with influenza. That's a good question.
Okay, thanks for confirming that. So we also have a question from Sergio Mohammed Usman, and he congratulates you for reaching 100% mortality stoppage with the CTAC, but he's wondering about viral shedding. Do you have any
Yeah, so
comment?
that's a really great question. And that is also on our list of things to study. We think that we could probably try that in the context of influenza in something like a ferret model where we co-house animals, because that is the other benefit of driving a strong mucosal response, right? If you are carrying less virus in the mucosal surfaces, then that probably means you're shedding less virus, which means we could potentially have an impact on transmission blocking, which isn't typically something that we study when we ask whether or not our vaccine is efficacious. Or I guess people study it more recently with SARS CoV-2. But yeah, we're definitely interested in those sorts of transmission blocking studies in the context of influenza and potentially SARS CoV-2, because that would be dramatically important sort of for public health in the context of designing vaccines for respiratory pathogens.
Oh, great, great. I see questions are still coming in for you. Zalma asks, considering your proposed molecular mechanism, do you think the mucosal immunity and protection will be similar? If the CCL27 was to be administered in the context of a protein vaccine, meaning both the antigen and the chemokine as proteins themselves,
That's
what
a
do you?
very good question. I think that it could work in the context of protein delivery if you had a way to be sure that your chemokine and your antigen stay in the same lymph node, right? You want to be priming cells in the presence of this chemokine. So I think it can definitely work in nucleic acid. It could definitely work with a viral vector delivery system. Protein might be a little bit more hard, and the concentrations that you need to get might be a little bit different. So when we give DNA plasmids, we're sure that cells at the site of infection are taking up both plasmids, so those cells are making both products. It's that relationship might have to be tweaked a little bit in the context of protein, but I think you could do it. I think probably the best way to do it in protein would be to deliver protein and CTAC intranasally.
Okay, okay, okay. Sounds exciting. So I think you've answered all your questions. Do you have additional questions from Ebony? Okay, no more questions. Thank you so
Thank
much,
you.
Ebony. That was very interesting.
Thank
And
you.
we're going to move on to our next speaker. So our next speaker today is Dr. Annie de Groot. She has served as the CEO and the CSO of the biotech company EpiVax for 25 years. Currently, she maintains her role as CSO and she continues to lead EpiVax as chairman of the board. She is a graduate of University of Chicago and Smith College, and she trained in infectious disease at the New England Medical Center and vaccines and immunology at the NIH. She has authored several publications, more than 228, and she has several patents, over 46 patents. In addition to engaging in a wide range of research at EpiVax, Brown, URI, and University of Georgia, she maintains an active role in two not-for-profits, Clinica Esperanza and the GAIA Vaccine Foundation. So today we have Annie talking to us about computational vaccinology. Over to you, Dr. Annie.
Can you see my screen?
Yes, please.
Okay, great. So
Right.
I'm going to give a slightly different talk from that wonderful talk we just had from Ebony, and I learned a lot from that and have lots of questions, but I'll send them to her separately. Very exciting work from the David Weiner lab as usual. So I'm going to talk about kind of the trajectory of computational vaccinology and how we kind of got to where we are right now, and hopefully give some I don't know, it's not advice, but just some background on how you kind of negotiate your way through the system to get to becoming CEO or CSO of a company and having an influence on the field. So I'm here to encourage everyone. That's really the point of this. So I started actually when I was at Smith College doing programming, Fortran programming, back when we had punch cards, and went on to the University of Chicago Medical School with a National Health Service Corps scholarship, and then did a post-medical residency at NIH because I was supposed to pay back that National Health Service Corps scholarship, and became a medical research training fellow at the NIH. I worked with Russell Howard, Michael Good, and eventually Jay Brzezowski there, and then then did an ID fellowship at Brown University, at New England Medical Center, and then moved to Brown University with my first assistant professorship in 1993. Some of the papers that I published at NIH are shown here, and I just wanted to point out that a lot of times what happens when you're a fellow, you keep on doing that work. So whatever you're doing now, you must love because you tend to keep on doing that work. And one of the things I was working on was HLA and immune response and T cells. And in this case, I was working on the Plasmodium falciparum vaccine program at NIH. Fast forward, we've developed some tools that help us design such vaccines. And so this talk will be focusing on the EpiVax platform. I won't be talking about our platform called AHRI, which is used for biologics. But the most recent paper is shown here. It's in Frontiers in Immunology online. So just really briefly, I'm going to talk about how we got started, the tools that we use, and some applications of computational vaccinology tools so that you can kind of get a flavor. I do have to give credit to the giants on whose shoulders I stand. And so really the work of Peter Doherty, who's now at, oh gosh, I forget, yes, it'll come to me. He's in Georgia, and Rolf Sinkernagel, who's also one of the founding fathers of HLA restriction of immune response, and really described to us how HLA has an impact on immune responses to vaccines. And so the reason why that was so influential for me was basically that it became clear that it wasn't just the whole virus or the whole virus protein, but actually peptides from the virus that were being presented on the surface of antigen presenting cells that were restricted by individual HLA that were really important and to immune response. And that was important because we could predict these epitopes using bioinformatics. And that made it possible to analyze vaccine antigens and vaccines purely by getting the genome sequence of the virus and feeding that into your computer. And when I was working at NIH, I was actually working with Jay Brzezowski, who in collaboration with Charles DeLisi and Hannah Margalit, had actually started looking at how do peptides from viruses get presented on MHC. So Doherty and Zinkernagel said MHC was important, but these guys were trying to figure out how that actually worked. And they didn't know that peptide epitopes were linear and binding in the HLA groove in a linear fashion. So they kind of started looking at the structure of the T cell epitope that was binding. They had an idea that the side chain variation had an impact. And here they're showing that there are some side chains that anchor into the HLA molecule highlighted in blue. So they called this the Twisted Helix, and we thought that was a great name for a punk rock band at the time. Further work was done by Roman Heach and Robert Urban in Strominger's lab when they saw that there was a peptide binding in the MHC molecule. It was called an indeterminate density. They actually realized that it was linear, and they started looking at how those peptides bound by eluting peptides off HLA. And they found that there were certain patterns. And here what I'm illustrating, but with the arrows, are the same side chains that bind into the HLA molecule that were first described by Brzezowski, DeLisi, and Margalit. So we now know that there are side chains binding down into the HLA binding groove. And those became what these guys, Heach and Urban, called motifs. So if you had a motif for binding, you could actually search for that in the sequence of the genome and use information about motifs to search a sequence in the computer. And that was really exciting for me. When I saw that, I realized that all of those sequences that were getting published could actually be fed into the computer, and we could start identifying motifs and predicting which parts of the virus could actually generate a T-cell response in both, mouse, human, didn't matter, as long as you had the motif for the HLA. And at the same time, what was happening is that all of the Craig Ventnor people were sequencing genomes like crazy. The first H. pylori genome was published. I put that on the door of my lab because I was so excited about turning this straw into gold, so to speak, figuring out how to make a vaccine for H. pylori, and that we actually do have papers on that. So what do I mean by motifs? When Rotschke and Falk and Heach and Urban eluted peptides off MHC binding MHC molecules that were presented by antigen presenting cells, they found that they could actually see that there were specific amino acids in specific positions. And so that enabled us to start building search functions. And truly the first search function I built was Microsoft Word Find, because with Find, you can put wildcards in at all of these positions that are Black and use those V and L motifs to search a sequence for potential epitopes. Now we don't use something that's quite so brute. We actually use C++ programming and Oracle databases, and we feed the data from mass spec and HLA ligands that are published in IEDB, and you can use all of that information to build the tools and get not just motifs, but actually the relative proportion of which amino acid binds to which position in an HLA molecule, and also which amino acids might interfere with the binding. So all of that information basically made it possible to consider the idea that we could go from genome to vaccine. And that's what we were doing at the TB HIV Research Lab at Brown University when I met Gabe Meister, who was both an artist and a computer programmer. And he suggested that we start building a program using macros in Excel to develop an automated program to predict these sequences in any genome of interest. And we also had the help of Bill Jesdale and eventually Bill Martin, who is the co-founder of EpiVax, to applying their brains to the problem. And once we started doing this, we started getting requests for mapping antigens. And I decided that we should spin that company out of Brown University because the commercial work was interfering with the laboratory research, so to speak. So we've been doing this for 25 years now, 26 years actually. And over the years, we've kind of made a number of different discoveries, some of which I'll touch upon today. And specifically, I'll be talking about EpiVax. EpiVax is an online tool that allows you to look for class 1, class 2 epitopes, analyze for homology, design vaccines, et cetera, in silico. And we back that up with a lot of in vitro assays and HLA transgenic mouse research. Because we had started this whole program, I was actually invited to a gathering of immunoinformatics experts, and you can see some of these people are fairly well known. Notice how few women were actually at this original meeting. I just want to point that out because it's a fact of life, right? But now we use AI and ML, that's integrated into the programs that we're using to predict even better and more accurately, and also to look at the impact of HLA on global, regional, and personalized immunogenicity, to detolerize antigens. I'll talk briefly about that. And of course, we can look at T-cell receptor analysis, something we haven't tackled yet, but we want to use that information to identify key targets and antigens. Now, we focus on T-cells. I think you heard from the last talk how important T-cells are to protection against COVID, because they were essentially measuring CD8 T-cell response. And of course, CD8 and CD4 are very important. CD4 helps with the generation of a high affinity antibody. So if you don't have T-cell activation, you don't get plasma cells, you don't get memory B cells, et cetera, et cetera. I don't need to tell you guys that, you know that. Also, T-cells support CD8 T-cell activity. So you need both CD4 and CD8 epitopes to produce an effective immune response. Because T-cell epitopes are linear, we can actually predict their binding, as I've described. We start by looking at the HLA binding pocket and matching that up to not a motif, but actually a matrix of potential amino acid side chain possibilities. So our tool is called EpiVax. It predicts MHC ligands or HLA binding, and we also have mouse and pig models. And we can do both class 1 and class 2 because that's both of those are very important to immune response. Now, I just think this is a really important study. Greg Poland did this study. He did a GWAS study in Minnesota, and he looked at why antibody titers vary. And I think this is really important because we forget this. We think that if 20% of the population has antibody, that means that each individual has a 20% antibody titer. That's not true. We have great variation in populations, and we actually have people who have really strong immune response and people who have weak immune response. And what he showed in this giant GWAS study was that the variability in the immune response to measles was due to HLA-DR. And that was the predominant signal in this giant study that he did. And that means that because different people see different amounts of T cell epitopes, that the strength of the antibody response is related to having more T cell help. So if you follow that theory out to its logical conclusion, more T cell epitopes actually drives a better immune response. Is that what Ebony's CCL27 is doing? Maybe generating a better presentation of T cell epitopes at the antigen presenting cell surface? That would be really interesting to figure out. Now, which HLA should we look at? Because there's so many of them. It turns out that a lot of the motifs that relate to the binding of the HLA, of the ligand to the HLA binding pockets, are conserved between HLA molecules. So you can kind of classify class 1 and class 2 HLA into families. And these are what are called supertypes or superfamilies. And there are many families, but you can condense the HLA-DR allele down into 9 distinct HLA-DR haplotypes that are important for binding. And the publications that describe this are on the slide. So that's what we use for class 2. We also have 6 haplotypes for supertypes for class 1 shown here. And that's basically what we use when we're doing global populations. But of course, when we're looking at individual immune responses, we use their individual HLA. So because more T cell epitopes is better for immunogenicity and antigenicity, you can see that we can predict now immunogenic potential of a single antigen by counting up the epitopes that are presented in a population or an individual. And that's what we do with our scale here. So here you can actually see that we've normalized all of the T cell epitope content to 1,000 per 1,000 amino acids, and you can compare antigens to each other. And what's really interesting about this scale is when we first started doing is that we realized that there were effective antigens that were on the plus side of the scale, and some maybe less effective antigens that were on the bottom of this scale. So here's some illustrations. Hepatitis B surface antigen is a great antigen, very immunogenic, practically, I think, 95% effective as a vaccine. Tetanus toxin, maybe less so. Is it possible that tetanus toxin is trying to escape antibody response by generating fewer T cell epitope responses? Maybe. And you can see that a failed vaccine, 85A for MTB, is actually -11 on our scale, and so is CSP. So you may be familiar with the RTSS vaccine, and actually that I have some side studies on that. We've looked at RTSS. We find that it's poorly immunogenic in our system, and that sort of correlates with a very poor efficacy in the studies that have been performed. Even though they bind the RTSS to hepatitis B, you still get a pretty low seroconversion and protective efficacy in these studies that have been conducted in Africa. And then there's a whole story about 85A. That was a big Gates Foundation project, which failed. So traditionally, we think about one antigen fits all. And so here's another example, spike protein and H5N1 also. Very good potential immune responses based on T cell epitope concentration per unit protein, and less effective for falciparum. And I also argue that the selection of the CSP sequence that was put into that vaccine is not the best sequence. There are better sequences circulating out there that could have been used. Now, there are other types of tools that we've developed, which are on the website, the IVAX website. I won't go into these because they're published. There's one tool for finding conserved 9-mers that can then be assessed for potential HLA binding. We've used this quite a lot for HIV vaccine design. We also have a tool called VaxCAD, or computer-assisted development of vaccines. And you can see that you could basically identify epitopes that are really immunogenic, but might not be identical in each of the strains that are present, say, in HIV or hepatitis C or influenza. So we can search for epitopes that are kind of in the same region and then build an immunogenic consensus sequence. And we've shown that those immunogenic consensus sequences are highly immunogenic in exposed individuals in flu, Zika virus, what's the other ones, HIV, quite a few pathogens. So VaxCAD puts these epitopes together and looks for junctional epitopes and then reorders the epitopes so that there's no pseudo epitope at the junction. Another key feature, just checking the time here, is the idea that you could identify Treg epitopes. And this came out because we were working on Treg epitopes that we had found in immunoglobulin in monoclonal antibodies. And so we were asked whether the peptides that were presented to Treg cells and T effector cells had similar binding propensity based on epimatrix. And yes, they do. So we reasoned then that the TCR face might be relevant to the discovery of regulatory T cell epitopes, because that might be what's driving the Treg rather than the T effector immune response. And that has borne out to be true. So in 2008, we discovered Treg epitopes in immunoglobulin G. These are peptides that have strong signals for T cells, what we call epibars, which are basically promiscuous T cell epitopes. These were highly conserved across IgG. They're even conserved across species. And so you can do experiments in mice using human T-regitopes, and they induce natural Tregs to modulate immune responses to whatever antigen you combine them with. So whether it's a tularemia protein or it's enzyme replacement therapy, you can modulate immune response to those antigens using these T-regitopes. Naturally, what they do is suppress T cell response in the lymphoid follicle, and they basically reduce antibody affinity and titers. So with Tregs present, you actually get poor antibody response to antigens. And that makes sense because what pathogens have then done is introduced Treg epitopes into their own sequences. So we actually looked for Treg epitopes that were peptides that were present in pathogens, that were highly conserved in the human genome and could be conserved at the TCR face with other Treg epitopes in the human genome. And in this study that was carried out by a graduate student at Dartmouth, we found that when we looked at 51 human viral pathogens, we found T cell epitopes that were highly conserved with human proteins, and those pathogens that were more likely to be commensal and live with you for a long time, EBV, for example, had more of them than pathogens that were likely to kill you quickly, like, say, Ebola. And so the presence of these Treg epitopes potentially modulates the ability of humans to make antibody responses to the antigen. So they should be removed. And in fact, that's what we did. First, we published this paper on what we call not immune editing or deletion of T cell epitopes, but immune camouflage, where Treg epitopes are introduced. You can imagine that for TB, parasites, lots of HIV, HCV, there are lots of Treg epitopes in those pathogens. And in fact, that is what we have found. So we built a tool to find these Treg epitopes called Janus Matrix. It looks at the TCR face. So it basically splits each epitope into the amino acids that bind down into the MHC, looks for the TCR facing residues, and then compares those to the human genome. And this is the graphic from that publication from the Dartmouth graduate student, Lu He, showing that pathogens that kill you quickly have fewer Treg epitopes than pathogens that like to live in your body for a long time. So please take a look at that paper. It was really fun to do that work. So we postulated that you could remove these Treg epitopes to improve vaccine efficacy. And so we started looking, for example, at hepatitis C, and we found that there were some really very impressive Treg epitopes in the highly conserved T cell epitopes that are conserved across subtypes of hepatitis C that might modulate immune response. And we also found some in H7N9. So you may remember this was a previous avian influenza. We found a Treg epitope that actually did in vitro modulate immune responses to other epitopes in that same virus. That might have explained why there was such a poor response to the H7N9 vaccine, the monovalent H7N9 vaccine, much less immunogenic than the H1N1 pandemic flu vaccine. So we then looked at that H7 epitope and we modified it. We introduced a promiscuous T effector epitope in its place. And just by changing 3 amino acids, we showed in a humanized mouse model that we were able to improve the antibody response. And we've subsequently shown in studies performed at the University of Georgia that these modified H7HA antigens could protect against live challenge in a mouse model, so a HLA transgenic mouse model. And also true for cancer. So really interesting recent work of ours is that we found these Treg epitopes in the mutanome of individual patients' cancers. And have been working diligently on developing personalized cancer vaccines by looking at the T cell epitopes that are present in individual mutanomes, and have shown actually that if you eliminate those potential Treg epitopes, you can predict who survives. This is checkpoint inhibitors, but we showed that we could actually differentiate survivors from non-survivors by looking at their mutanome and showing that they had more epitopes and fewer human-like epitopes, they were more likely to survive. So we built a tool called ANSWER for personalized cancer vaccine development, and that tool is still out there. We're collaborating with scientists in Japan. Last point I want to make is that if you have all these tools, can you actually predict which vaccines would work? And so we developed a tool called EPIC, where we can look at the epitopes in a vaccine, say your COVID Wuhan vaccine, and see if it will protect against Omicron, JN1, the variant of concern. And you do that by basically looking at the conservation of the T cell epitopes with your vaccine. Obviously, 100% conservation would work if you had each strain being a vaccine. But since we have vaccines that are based on single variants, usually we get pretty good coverage. You can imagine if this were the variants of concern going around the time frame here, that you would actually get a drop off in the protective efficacy of a Wuhan vaccine for the, say, Omicron variant. And indeed, we did find that. This is just an initial study where we looked at the Wuhan versus the Omicron vaccine in terms of protection against all the variants. We did pretty well with the Wuhan strain, with good coverage of T cell epitopes up until Omicron. And it took us quite a long time to make the Omicron vaccine, but when we finally did, this is the actual strain that we use. We can see about a year's or less than a year's worth of protective efficacy, and then we start seeing a drop off when XBB shows up. So T cell epitope coverage is critically important for vaccine efficacy, and we've actually now commercialized this product, EPIC. It's being used in the swine viral pathogen field to select the best match vaccine for circulating isolates of PCV2. So last but not least, we have other tools. AHRI is a tool we use for biologics, and we're currently at actually 4 million sequences per year being evaluated in this tool. So I've talked about all these things, and I just wanted to say that I'm excited to work with my company to continue to improve these tools, and hopefully we can contribute to the integration of these tools into the field of vaccinology, enabling vaccine and biologics development to progress with greater speed and efficacy and safety for people and animals in the 21st century. So I'll stop there. Don't know if we have time for questions.
Thank you. Thank you so much, Annie. That was very great and informative. And we have time for a few questions. So if you have questions, can you please raise your hand or put your questions in the Q&A box? We already have one question for you by Sergio. And Sergio says, I learned that EpiVax use only human MHC alleles. What will you suggest for predicting chicken pathogen T-cell epitopes?
Chickens are difficult. So we actually are looking into that right now. We've actually done pigs. And so pigs, I will be going to a meeting actually this weekend in Chicago on swine pathogens, where I will be talking to collaborators who are interested in using the pig-focused vaccine design tools that we have. We basically substitute the MHC of pigs in for the human MHC in the IVAX toolkit, and we can use all of the tools that we have for pigs. Have we done that for mice? Yes, because we were forced to use mice in our preclinical studies. So we have mouse, BALB/c, and BLAC6 alleles as well. And we have looked at chicken. The problem with chicken is that the MHC binding groove is very shallow, so it's a little more difficult to figure out what actually binds there. But I think there are some recent studies showing that you can actually use human MHC binding information to build chicken predictions. And so we've started looking at that, and we'll be hopefully making improvements in the future. I am committed to making better vaccines for animals, because obviously for food production and the vast majority of the world is dependent on keeping animals pathogen free. And so vaccines are the best way of doing that. So we're going to be continuing to work in that space with the IVAX toolkit. Once we can develop the MHC prediction models, then we should be good to go for chickens.
Oh, great, great, great. So Anki wants to know, okay, Anki first of all acknowledges your great presentation, and they are wondering whether you could design an ideal T helper cell epitope that binds well with a whole range of HLA molecules, and this will be used as a universal epitope with different B cell epitopes. Is this something that is possible?
Yeah, you could. I think it's interesting because I think for immunogenicity, that reaction that drives an immune response is really local. It happens in the lymph node. The B cell or the antigen presenting cell are presenting the T cell epitopes, and usually the B cell that's making the antibody has to be right nearby, or the CD8 T cell that's going to be killing the cell has to be trained by a T cell. So it's local, local, local. That's basically very important, these interactions between individual cells. So you could develop a vaccine that uses a universal T cell epitope. And in fact, people have tried to do that with the Padre epitope. You may be familiar with that. It's an epitope that was developed by Alex Sete from La Jolla Allergy and Immunology Institute, PADRE. It is a promiscuous T cell epitope, binds to multiple HLA class 2 alleles. It hasn't worked out that well in the vaccine space. I don't know why. I think that there's more to it than just a single epitope. So that I guess that's not a very helpful answer, but we like to see more T cell epitopes in our antigens. And so we actually built in more epitopes into the H7 and 9 antigen. We added H3 epitopes. And in studies that have been published, we showed that that was even better than just adding one epitope. So more epitopes is better, I guess, is the message, not just one universal epitope.
Okay. Can I ask a general question just before we round up today? And this question is for an early career researcher on how you manage to spin up a company from your lab and still do research and still have the company. Like, how does that work?
I think it's really important to do research when you build a company. So we're very science focused at EpiVax. And one of the things that's important, I think, when you're building a company like this is make sure that you can support yourself. I know that sounds crazy, but being able to generate revenue, as we did initially, we generated either grant revenue or commercial revenue when we started the company, gives you the ability to stay independent. You do not have to sell yourselves to a board of investors, and you can pursue the ideas that you think are most interesting to you. That makes it possible for you to continue to be happy about going to work every day, as I am 26 years later. I'm still excited about going to work. There are things we're working on that we're publishing this year that I'm very excited about. And we don't have investors. We don't have investors because we've been able to support ourselves and grow to 44 people over the years by being very fiscally conscious and responsible, but also by focusing on the science. And so I think those are the answers to that question.
Okay, we need to round up, but I see a question. So can you quickly talk to us about how people can have access to your training courses, or like can early career researchers?
So yes, when we had NIH funding, we had a big U01 grant when I was at University of Rhode Island after Brown. And we did used to do in-person trainings. Now what we do, and we talked about this before we got on, right, Lara? We have trainings that occur at the International Society for Vaccine meetings. And so the next one will be in South Africa. I will be there. I'm hoping to integrate training into that project, into that session in Cape Town. So hopefully I'll see you all there.
Thank you. I think that was just the right ending for this webinar. And that is enough encouragement for everyone attending today to join the ISV. And also, if you're an early career researcher, please join the Next Gen Vaccinology group using the QR code, or send us an email. And yeah, we look forward to seeing you in our next webinar. Thank you, everyone. And thank you to our speakers for today and for everyone that attended today. Thank you.